Welcome to the Cannabis Cultivation and Science Podcast. I'm your host, Tad Hussey of Kiss Organics. This is the podcast where we discuss the cutting edge of growing from a science-based perspective and draw on top experts from around the industry to share their wisdom and knowledge. Our guest this week is Professor Yoram Kapulnik. Professor Kapulnik is the Executive Director of the U.S.-Israel Binational Agricultural Research and Development Fund, also known as BARD. He is the former Director General of the Volcani Center in Israel from 2011 to 2016, and prior to this position, he served as the head of numerous institutes within the Volcani Center. Born and raised in Israel, Professor Kapulnik received his doctorate in soil microbiology from the Hebrew University and as a Fulbright Fellow completed his post-doctoral work at the University of California, Davis. He worked for more than 25 years as a professor of plant physiology and biotechnology at the Volcani Center, Israeli Minister of Agriculture. His early work focused on the study of plant-microorganism interactions, mainly in plant roots, and their impact on crop productivity. Later, he concentrated on identification and utilization of natural products produced by numerous plant species and their health benefits to humans. Professor Kapulnik holds 14 patents, trained more than 65 graduate and postgraduate students in his lab, and has published more than 170 articles in his field in international scientific journals and edited 3 books. Now on to the show. Dr. Kapulnik, thank you for coming on the show today. I really appreciate it.
Thank you very much, Ted.
Yes,
I'm really waiting for that.
Well, I have a whole bunch of questions for you. I think your research is really interesting. But could we start off letting listeners know a little bit about your background and who you are?
Okay. Actually, I'm a scientist. I obtained all my degrees here in Israel. And towards the end of my when I graduated from my PhD program, I went to the US and I spent 2.5 years in UC Davis as a postdoc in the agronomy department. And then I went back home to Israel and I took a position in the agricultural research and development. This is actually and governmental institute, we call it the Volcani Center, and nothing about Volcano, but this is the Volcani Center, which is celebrating now this year 100 years of existence. Much before the state of Israel was formed, people here try to develop agricultural knowledge in order to be able to supply their food. So I joined the field crop department, and I served as a scientist for 30 years. Along my research, I took a lot of administration position, and the last one was I was the head of the ARO, the Agricultural Research Organization, the Volcani Center, which consisted out of 1,200 employees, scientists, technicians, and students. And in this institute, I dived into what we call it Rhizosphere biology. Rhizosphere is the closed niche around the roots, which is actually the soil which is surrounding the roots. And there's a lot of biology is happening there. Not just biology, chemistry and physics, everything is actually concentrated to the first 1 or 2 millimeters of the root surfaces. And I studied a lot of plant microbe interaction as well as roots physiology. So this is my background, the rhizosphere and biology. And during my academic year, I taught in the Hebrew University a course. This is a graduate course, which is root structure and function. So in a way, we hold the issue from both ends, from the plant side and from the soil side. And the arena is the rhizosphere, as I just mentioned.
Well, you have a fascinating background. I study, on my own time, a lot of the rhizosphere interactions that you're talking about, purely for my own education. And it seems like such a complex interaction between microbes, their exudates, the root exudates, and the mineral content of the soil. Before we dive into some of this stuff, some of the questions I had about mycorrhizal fungus, can you kind of summarize the symbiosis of that relationship and how it works? The basic version that I would put out there is the plant's releasing exudates into the soil that the microbes then consume and cycle nutrients back to the plant, in addition to breaking down the organic matter and other minerals that are existing in the soil. And the plant kind of controls this interaction based off of the exudates that it's releasing and the chemicals it's producing itself. Can you kind of elaborate a little bit on that, or talk about that relationship, or even correct me if I stated anything wrong?
I think that you were right generally, but just to the sake of for our audience, they need to understand that there is not kind of a passive, kind of a leaking system that incidentally the plant is losing one molecule or two molecules. We are talking here about a massive phenomena. About 30% of the photosynthate, I mean, all the carbohydrate that the plant is accumulated through the photosynthesis, is actually find its way at the end of the day, or along the whole relationship in the soil, in the rhizosphere. And so this is a very rich environment relative to the soil itself. You find there in the root exudate a lot of sugars, of course, but also vitamins, amino acids, nucleic acid molecules like DNA and RNA. I mean, whatever you think of the plant content or the cell content, so you can find it in the rhizosphere in a certain concentration. And that's actually drive the whole biology, because you have consumers of those goodies which are being supplied by the plant, and there is competition to reach them. So it's a very, very active region, and we have to remember that the tip of the plant, the tip of the roots is not like a more mature part of the roots. The exudates or the chemistry that you'll find around the root tip is totally different from the elongation zone or from the branch zone. Each one of them has its own or different exudation mechanism, and that actually influencing the biology around it. And once the plant, now if we take it one level further, there is a lot of saying there. I mean, talking about chemistry discussion between the plant and the microbes. And some of them can even reach farther and penetrate to the tissue, to the plant itself. Of course, through the evolutionary process, that you need really to provide something good that the plant will allow you, to break in and to enter to the root itself. So we find a lot of several groups. Many of you probably familiar with the nitrogen fixation system. These are rhizobia, which penetrating to the legume plant and really fix nitrogen. So in a situation where the plant has enough light, enough other nutrients, but a shortage in nitrogen, actually this biological system can really feed the entire needs of the plant. And that's something which is very, it's being used in agriculture, like peanuts and soybeans are being inoculated, and we can we were able to tie them in to our conventional agriculture. Another group of microorganisms, these are mycorrhizae. Now, the mycorrhizae, we have at least two distinct groups, some of which are not really penetrating to the plant itself, These are the ectomycorrhiza fungi. It's a lot of plant species which belong to that. And most of them are colonizing perennial, like trees and shrubs.
Conifers.
And excuse me?
Yeah, so I think of conifers when I think of
Like
ectomycorrhiza.
exactly like conifer, a lot of mushrooms, and there's a lot of ectomycorrhiza fungi which we know, which has some gastronomic effect, and in a way, now I would like to drag the discussion to the other group. The second group are those type of fungi which has the ability, which with the evolutionary process were able to colonize the tissue itself. So if you look on the roots under the microscope, you'll be able to see the hyphae of the endomycorrhiza fungi actually spreading in the root tissue. Not only that, but some of these fungi, not all of them, can really produce some what we call it vesicles. The vesicles are storage bodies. So not only that the fungi can grow in the rhizosphere, and like you describe very right, that it can absorb some nutrients and minerals and translocate it to the tissue, to the plant itself. The fungus can even store it for a while in the tissue itself. In a way, it's being protected, and it is ready to use in the roots itself. In some of the cases, the plant was able, even kind of, if I describe it simply, to fool a little bit of fungus and to, in a way, to consume some of this storage without permission. And that's this is kind of, we call it symbiosis, but in a way, each one of these parties is trying to make living out of this situation. So in a situation that the plant is suffering from lack of some minerals, and these minerals can be somehow collected and transported to the tissue itself, in exchange, the fungus is actually receiving a lot of the photosynthase, which are coming from the leaves to the roots. So, well, the fungus cannot do it. I mean, if we take the endomycorrhiza fungi and we try to feed it artificially by any type of sugars, we probably will not be able to do it on a petri dish in the laboratory condition. I mean, the fungus really need vigorous and roots which is alive. And then comes another layer of question: how comes that the plant is allowing the fungus to penetrate and to spread out in the tissue itself? While when a pathogen is trying to do the same, the plant is responding with plant defense response. And we know that the plant has the ability to defend itself. And the question is, why mycorrhiza fungi are able to spread in the tissue while other pathogens sometimes are being blocked. In many cases, they are being blocked, or the plant is trying at least to fight back and to prevent that penetration and colonization. And, this is a long story, and we studied these processes for many years. And to make a long story short, I can tell you that somehow the mycorrhiza fungi, they are able to turn off the plant defense response, like kind of this is type of conversation, telling that the plant, listen, guys, here comes the good guys. Don't aim your ammunition against us, because we are at the end of the day, we are trying to help you. We, the fungus, try to help the plant, and that's the benefit of the plant. So, I described it a little bit, and you're right. The rhizosphere is a very interesting and active region, and some of those organisms were able to identify a unique mechanism by which it will be justified to really to create that symbiosis, what we call it, for the mycorrhiza and rhizobium, which I just described now.
Yeah, that was great. I never thought of it as potentially in the same way as I would a pathogen, but that makes a lot of sense that it would need to somehow shut down that plant response as that sort of pathogen response. And you mentioned some of the benefits around this is it's a storage of nutrients. It provides more nutrients. It extends sort of the root system or the ability of the root system. What are some of the other benefits of mycorrhizal fungus? Because one of the main ones we always hear about is phosphorus uptake. What do you think of when you think of that association, though, in terms of the plant benefit from it?
Okay, so first of all, just to make it much more, well, it's not just phosphorus, of course. Here we're talking about most of those elements which are not moving easily in the soil solution. You know that in the soil we have the physical structure, and we have the in between those particles we have the water which is moving. And a lot of minerals are being dissolved there. But like iron and iron are being and phosphorus are being attached to the solid surface and are not easily moved with the soil solution. So first of all, mycorrhiza helps to the plant to reach out and to receive more of those elements. This is the first thing. Another thing which is very clear to us that something in the physiology of the roots is altered by the existence of the mycorrhiza. We know that the resistance to plant nematodes is much higher when you have a mycorrhizic plant. Now, another benefit which is well known at the plant level is there is kind of an enhancement of the photosynthate. Which, well, in a way, the mycorrhiza is charging the plant, but in the same time, it's helping the plant to gain more photosynthesis. How it's being done? It's being done by altering a little bit the very simple and unique molecules. We can call it hormone-like molecules that are moving in the plant itself. And that signal is reaching the leaves and influencing the leaves itself. It can influence the evaporation by stomach closure. The plant is not losing so much water, but in the same time is enhancing the photosynthesis. If we'll move out of the plant and we'll go back to the rhizosphere, something which is really unique about mycorrhiza, and this is true to endo and ectomycorrhiza as well, is that hyphae, the fungus itself, which is somehow growing or spreading away from the plant, the fungus itself, the hyphae is being exudating also some molecules. Some of those molecules are enhancing the aggregation that's actually improving soil properties. If you have a small aggregate you have in the soil, so the water intention and the structure of the soil is much better. So mycorrhiza helps the plant tremendously in this level. And last and not least, now we are talking about communication between nearby plants. That hyphae, which is connected to one of the plants, it can reach out to the neighbor plant, to the nearby plant. And we can see a lot of signals and even some moths which are moving by the mycorrhiza from one plant to another plant. That's very important to when you're talking about field crop, because in the field crop, the plants are so dense, and sometimes, let's say, water is not reaching out at the same level to every plant. And the mycorrhiza is there to somehow to smooth the differences between the plant and to create kind of a more unique population of plants per growing area. So this a lot of things. There's more and more things which can be added to that, but I think that I mentioned the key or the most important ones. The fact that here I need to say something which in a way is contradictory to what I said a couple of minutes ago. I told you that in order to spread into the plant, somehow the plant defense mechanism should be shut off. But that reduction in the ability of plant defense response is only for a short while. It turns out that the plant which is mycorrhizic is when a pathogen is reaching in just after colonization with mycorrhiza, then the plant is responding much more aggressively in terms of altering the plant defense response, fighting back when the pathogen is trying to colonize the roots. So those type of changes on the physical side, but also the chemistry and the hormonal side are very, very clear for that. So the plant which is mycorrhizic may be a little bit more sensitive at right in the next vicinity to the penetration attempt of the mycorrhiza. But after a while, I'm talking about half a day, everything is actually retrieving back, and the plant is not exposed by the mycorrhiza to a situation that it will be weaker. It always created a stronger situation later on.
That makes sense. So overall, you're getting a better, you're getting more ability of the plant to protect itself against pathogens when it has that mycorrhizal association over time.
Yeah, but we know from different crops that even the flowering is you can get an earlier flowering on the plant. You need less days to flower set, which is very important sometimes in agriculture, especially in a region which are dry region. You really want to finish quickly your crop. So it's not a question only of enhancing the mineral status or the nutritional level of the plant. Is a question of more kind of a hormonalic effect, which set the flowering a little bit earlier, and that's very not only earlier but more uniform even. We can see it on tomato and cucumbers. All of them, you can see the flowering coming earlier.
That's amazing. So you're saying earlier flower set, and did I hear you correctly when you said that a mycorrhizal association actually affects the opening and closing of the stomata?
Yes. Well, we ask ourselves, how comes, if the plant is, when you close the stomata, in a way, you're not losing water if you have, high temperature or dry conditions. But in the same time, you block the plant. CO2 CO2 is not getting into the plant. So the machinery of photosynthesis is, in a way, is slowing down because you don't have enough CO2. So what's really happening there is that under dry conditions, somehow the mycorrhiza is allowing the stomata staying a little bit more opening on the stress when the stress is how we call it when you initiate the stress on the plant, and when the stress is over, and when we are talking about recovery, the mycorrhizae plant are able to open their stomata earlier than plants which are not mycorrhizic. So in a way, mycorrhizic plants are using more effectively the light hours, or more effectively the photosynthesis process, even though there are some environmental stresses which in a way imposing stomata closure. With mycorrhiza, you do it a little bit more smarter. And by that, you're allowing the plant to have more CO2 and to fix carbon when non-mycorrhizic plants cannot do it.
That's amazing. I've never heard of that. So that I learned something new. I really appreciate that. That's amazing. I want to talk more about specifically some questions around mycorrhiza, but I'm worried that people listening to the podcast may not really understand that when you're talking about endo and ecto mycorrhiza in relation to plants, you're talking about essentially a family of organisms. It's not one particular species here. So I want to just kind of highlight this. So in terms of let's say a tomato crop or a cannabis crop, that has a specific endo mycorrhizal relationship, not an ecto mycorrhizal relationship. Because I see a lot of these products out there here in the United States that'll have, and I pulled one up to give you an example. It may have 83 propagules per gram of an endomycorrhizal species, and then 187,000 propagules per gram of an ectomycorrhizal species. And that creates a total number that's much higher. But that ectomycorrhiza doesn't have a relationship with our cannabis crop or our vegetable crop. Is that correct? Are there any other associated benefits to having these high levels of ectomycorrhiza in the mix?
First of all, you're right, and from my own experience, I don't have a reason to believe that ectomycorrhiza fungi do have a significant effect in cannabis, but I never tried myself. The only thing that I can tell you, and I should start it from another thing that probably your audience will be astonished to learn, that about 85% of the plant kingdom is being colonized with endomycorrhiza fungi. Here I'm referring to endomycorrhiza fungi. And in order to raise these fungi, the endomycorrhiza fungi do need a plant which is alive. And we are lucky for the ectomycorrhiza fungi. Remember, those creatures which are not penetrating to the plant roots, for them, we are lucky to have a laboratory technology that can allow us to grow the mycorrhiza in laboratory condition, and you don't need the plant, and you don't need the whole relationship and to maintain this relationship. So it's much easier to raise the ectomycorrhiza under biotechnological means, laboratories. And it's very tough to grow the endomycorrhiza. Not many labs can do it. That's why you'll find less propagules or less spores of endomycorrhiza in those products. And that's was our ultimate question in the beginning. How? Because we try to study this association, and we try to purchase some inoculum. And we realized that we cannot find it, definitely not in Israel. And the one which was on the market was so expensive. So that's why we dive in order to find a way that will be a practical way and will give us a high number of infected propagules in the inoculum. But you are right in the sense that for tomato, I'm sure that ectomycorrhiza doesn't have for soy, for corn, ectomycorrhiza fungi do not have, as far as I know, a significant effect on plant development. While the endomycorrhiza, those who penetrate to the root, do have a significant effect, and this effect can be created in a matter of days. It's not months or seasons. It's a matter of days, and it's a significant. So if a small endomycorrhiza propagules, you'll have spores or other means of propagules, then your inoculum will be better as long as it comes from endomycorrhiza fungi.
So that was great. So in terms of endomycorrhizal fungus, there's a lot of different species out there.
Okay, sorry. I didn't answer this. Yeah, I didn't answer. Sorry about that. It's known. I mean, we had a collection that we brought to our institute from so many places around the world, and we had the collaboration with different scientists around the world. And we realized that some of those strains are better on a given plant species, and they are not that efficient when you go to another plant species. And so we had to find the right blend to a given plant species in order to maximize the effect. I'll give you two examples. One of them is a fungus that we brought over from the Far East, and that fungus was able to be very effective under low pH, more acidity conditions of the rhizosphere. Because we had in Israel a lot of basic soils, and we were able to isolate some local species here, and all of them were adapted to high pH. So, if you really want to create a good product, you should try to have kind of a blend which will. You have two options. One of them is to get a super strain. A super strain that will work effectively under different conditions, or you can go ahead and have multiple strains, each of which will have some beneficial characteristic at a given like drought or flooding. Some of the strains which we are isolating in the desert, when you're going to expose those strains to flooding, they will not make it, which means the strains that we were able to isolate, let's say, in the Israeli desert, will never make it with rice in those pounds of high level of water above the roots. And that's, I think, two examples which show you that there are different species. And now the industry, they have a wide range of different species, and they can find the right strains or the super strain. And the super strain sometimes is better. It's easier to produce. You don't need to bring along the ectomycorrhiza counts for your product. You can depend solely on the endomycorrhiza, and that's something which is appreciated by the growers. It's the price coming to the price which is manageable. If you need to raise, let's say, 3 or 4 strains of different, for your blend, different species, of course, it's double or triple the cost of the product. While when you have in hand a super strain, then it would be much easier to produce and to provide it to the growers in a fair price or cost.
So essentially, this super strain will work over a greater range of pH, greater range of water moisture content in soils, have just a more persistent ability to infect the plant. That makes a lot of sense. Now, the one that's the most popular out there that I've seen is Glomus enteratices. I believe it's referred to now as Rhizophagus irregularis. In
Yeah.
terms of like nomenclature, it's hard when scientists change names on us. But is this particular strain going to be the same if it was produced, say, at a lab in Canada as compared to your lab, say, in Israel?
Well, although both of us are using Glomus enteratices, I will use the old nomenclature. We have different strains. The one which we isolate here are coming from, from the desert, high temperature, low humidity. I mean, these are superstars which were able to make it along the evolution. And we very persist in a way to take those strains over the way that the way that we are producing it, to take them through very difficult times. So it helps us later on to maintain the vigors of our strains. The ones which are being produced under tissue culture condition, they are in terms of they are producing a lot of spores per unit of time, and I mean it's like we know it. They got used to that environmental condition, that everything is being provided in optimum, and as long as you don't tip the growth condition, which is a little bit, more tougher, then you probably can get the benefit out of it. But in some cases, and it's happening to all of us, irrigation schedule is a little bit changed. There is a kind of bumps in our cultivations, and you really want something that will be able to overcome those bumps, something that can iron those wrinkles in the growing curve, if you want. And that's why we decided to stay with this more vigor strain.
So what you're saying are these strains are not necessarily the same, and some of these tissue culture strains essentially grew up in environments that are essentially ideal for them. And so when they get put in a situation that's less than ideal, say in a grower's soil, where you might have more fluctuations in temperature, water conditions, and things, they may not be as vigorous as something that was more adapted to those conditions.
Yes, exactly what I meant.
Okay. I just want to clarify and make sure I'm understanding too. So, I want to talk a little bit about the persistence in soils, but before we do, can you clarify some things? Because I know some listeners, I saw a picture the other day of some really healthy root hairs online, and they were claiming that this was a mycorrhiza. And it was a vegetable root, so I knew that it was an endomycorrhizal plant that they were showing photos of the roots of. Now, my understanding is you actually have to stain the roots, and looking through a 80 times magnification like a jeweler's loupe is not going to allow you to see if your plant actually has that association. How can a grower determine if their plant has mycorrhizal infection? Or can they really?
I'm very pessimist that first of all, under lab condition, it's very easy to see the mycorrhiza. You have to stain it. So we are using a specific dyes, mm-hmm. which are bound to the hyphae of the fungus, and they cannot be bound to the root itself. So in a way, you can see the hyphae very clearly. If you don't stain the roots, you cannot see the mycorrhiza.
Perfect. That answers the question.
Yeah, that's very clear.
Okay.
Just looking at the root hairs will not tell you much. Now, if you have nowadays, scientists actually they learn more and more. Now there is specific molecules which is being accumulated in the leaf, and when you have mycorrhiza, if you have colonization down in the roots, that molecule is accumulated in the leaf up there. But it's really required more complicated and sophisticated chemical analysis and machinery. So unfortunately, not now, but I hope that we'll have in the future also some kind of a kits that will tell us exactly, you can squash leaves in against, a small plastic, and then have a couple of drops in, and it will change the color and will tell you if it's mycorrhizic or not. Unfortunately, it's still a dream. Well, some people are working on it, but it's still far from application.
Sure, that's really interesting. Thank you for clarifying that.
Yeah.
So you mentioned that the infection takes a period of days. What's sort of that range of time? And along those lines, are we really seeing results in a fairly quick crop? Like, let's just say lettuce versus cannabis. Is there any real benefit to a mycorrhizal infection in some of these faster crops?
Definitely yes. If you have a spore, let's say, there are a couple of ways to get the mycorrhiza into your root system. One way is to have a spore, which is it's like a seed in terms of, it's not a living organism it's a living organism, but it's in a dormant stage. Mm-hmm. It takes about 5 days from the moment that you expose that spore to the rhizosphere. Please listen very carefully. It's not from the moment that you wet the spore. When you expose the root to the rhizosphere and the spore is being exposed to a molecules which allow it to break, to break the dormancy, and the hyphae is emerging from the spore, and you can see penetration to a tissue, to the roots. Again, not to every part of the roots, only at the young stages of the roots, at the root tip. It's around 5 days. If you can see colonization at the more mature part of the roots after, let's say, 2 weeks or something like that. But definitely, the mycorrhiza can colonize the plant and can be established in a matter of days, or we can say after, like 3 weeks, you should start. If you have the right condition, you can start seeing some differences between mycorrhizic and not mycorrhizic plant. And that's it really depends on the growth condition of the plant. When the plant is more stressed, then the effect of the mycorrhiza is more obvious. It can be distinguished very easily. But we don't want to get to such a stress condition in our crops, in agriculture. So that's why it will take a little bit more. But it's in a matter of 4 to 5 weeks, and definitely you can see some changes. Definitely, if you do tissue analysis and you look for phosphorus and iron, you'll see manganese. You'll see high concentration of those elements in the plant itself, and it will start affecting. Now, the question is how bad the environmental condition is. If your plant again is a very optimal condition, nothing is missing there. You have the right wavelength, the right humidity, irrigation is perfect, aeration in the soil is perfect, then you might not see even the effect of mycorrhiza because everything is perfect. You have to bear in mind that relationship with the fungus is costly because the plant need to invest and actually to put aside some of the photosynthese which is being accumulated in the plant. Now, if the plant is not suffering and you have everything, so why to invest in something that will not do any different? For many crops, we say that it's like an insurance policy. You put it in this mycorrhiza just in case if something will happen, if something will break, if something unexpectedly will appear there, and then you have the mycorrhiza to help you out in that stress. And that, I can even use the same the same saying again. If you have a wrinkle in your growing protocol, the mycorrhiza can iron it, and it will not be that devastating to the plant. And the plant will not suffer so much from that mistake or that change in the environment or that unexpected situation. So in a way, mycorrhiza, sometimes if you have excellent growth condition, it will be very hard to see the difference. But you know that always there are some time points that we don't know, but something is being created there that or some regions along the roots. I mean, you have irrigation at the top of your soil, and down there the roots can be exposed to to drought. Not only that, but you have probably overwetting, and your roots are being exposed to anoxia and not enough oxygen. Again, mycorrhiza can help you out, and that's the benefit of having mycorrhiza in your crop.
Well, I think in reality, in the real world, in normal growing conditions, even with controlled environment agriculture, there still are a lot of plant stresses. No one is growing in a perfect, optimal plant conditions for the entire growth cycle. At least none that I've ever seen. This makes a lot of sense for them to form this relationship. But one of the things you touched on here was that if the plant has perfect conditions, it's not going to form this relationship. I've heard that in association with high phosphorus because the plant says, I'm getting all the phosphorus I need. It's already plant available. I'm not going to bother with this mycorrhizal association, even though there's other benefits to the mycorrhizal infection, like you just stated. So what are some of the best practices for using mycorrhiza, endomycorrhiza? It sounds like it needs to come in direct contact with the roots. It can be directly in the rhizosphere itself. We want to apply it early on in the plant's life. I would think at transplant. What do you see? Avoiding high P was the other one that I wanted to mention. What are some of your recommendations?
Well, from my own experience, as soon as you apply the mycorrhiza, is better. If you really want to reach those roots, the young part of the roots, which are actually those are the best location to colonize, you should have the mycorrhiza at a close vicinity to those locations. If we tried a couple of years. I can tell you a lot of stories, but we try to apply the mycorrhiza by drilling some holes to a more mature plant. And you have to imagine to yourself how much of the root surface really was able to enjoy the mycorrhiza, the existence of the mycorrhiza. And that's created a situation that the volume of the root system, which mycorrhiza can have a contact with, is very small. So the benefit to the plant would be also smaller. So, well, I still remember that, having saying so, I was involved in an experiment in the Kew Garden. In the Kew Garden in England, there are some plants which, very old plants, and that we try to help them because they were very old and they were not really in a really old plants. This is the right way to say it. And if you really want to have mycorrhiza there, so what we have done, we injected mycorrhiza around the trunk, trying to reach some of the roots and allowing that old plant to enjoy better the growing conditions. So, we tried also in Israel, in the cities. You have some trees that are not actually receiving the right condition and are suffering from soil compaction. So we try to inject mycorrhiza. But we saw some benefits after a while, and that's the type of experiment. It takes even half a year until you see something. If you really want to enjoy mycorrhiza in the growing season, make sure that you have it right there in the right position. And I know that there are some recommendations to put mycorrhiza on the soil surface. So, I can tell you again, it will take ages to those infection units and mainly spores, to a spore, which is it's a big structure, to really go through soil layer and to reach the root tip, which is sometimes 10 or 15 centimeters away, is ages. It's ages. You'll not be able to make it in the same growing season. So as soon as you have the mycorrhiza around the right region on the roots, then I'm sure that you'll be able to colonize better the entire root system. And also, maybe it's worth to mention something. When the mycorrhiza is penetrating, the hypha is penetrating to the root tissue, the next stage or the next step for the mycorrhiza is going and looking for something to eat, to get more photosynthesis. Where you have more photosynthesis when you're entering at the root tip, you have to migrate or to be expanded or grow upwards. So the direction of the mycorrhiza is from the root edge or the tip to the more mature towards the shoot. Okay, so it's going upwards and not downwards. And if you have a mycorrhiza and you apply it on the top, it barely reach the root system, and you'll not be able to colonize the entire root system. And then you'll blame the mycorrhiza that it's not working. Well, it's not working because it's not being spread downwards. It's being spread mostly upwards. So as soon as you put the mycorrhiza, when you have almost most of the root system around it, you can make sure that more of your root system will be colonized by the fungus.
That's great, because you see these micronized products, mycorrhizal products here in the United States, and really you can't micronize a spore. You're micronizing the carrier for the mycorrhiza, and even then, like you mentioned, trying to get it to work its way all the way down to the roots is going to take a long time if it even makes it. So I would just recommend to listeners that you follow this advice and apply it directly to the root zone if you have the ability to do that with a plant. So that's great. One other question I had around this idea of application is, I see a lot of products that have trichoderma and mycorrhiza in them. I've heard from some scientists that this is a symbiotic relationship. I've heard from others that due to the aggressive nature of trichoderma, some of the speculation is that the trichoderma is the benefit that a lot of these people are seeing and not necessarily mycorrhizal. because the other issue there too is the relationship will be like 1,000 to 1 in terms of trichoderma to mycorrhizal spores. What are your thoughts in regards to trichoderma and mycorrhiza?
Well, in our hand, we tried it because the trichoderma was or enter to the practice, to the nursery practice before mycorrhiza here in Israel. So we had to show compatibility to see if what's really happening there, and maybe the trichoderma would somehow eliminating the mycorrhization of the roots. And in our hands, we could see them living side by side with no significant reduction of in mycorrhiza. When you have a full trichoderma treatment, the recommended treatment. I'm not I don't have any experience with those products that are being sold in the US. But the mycorrhiza the trichoderma, which is being sold here in Israel, I'm sure that it's not harming the mycorrhiza. So that's my own experience.
That's great.
If you are using, in a way, those strains that are being right now are being used by groundwork. Some of those strains came from our collections. They can survive what a co-inoculation with no much of effect.
That's wonderful. That's really good to hear. How long does mycorrhizal fungus persist in potting soil? So if we were reusing our potting soil, I assume that there would be potentially some propagules in there, hyphal fragments, spores. Do we need to be reapplying mycorrhizal fungus every cycle in situations like that?
If you have a good population in your soil, and it's not just to have them, you have the right strain in the right concentration, probably you may not need to add more mycorrhiza. However, that's not always because sometimes you check for mycorrhiza in your soil, and then that soil would go to the warehouse and will stay there for a while. You have to remember that in order to maintain mycorrhiza, you need a living tissue. You need the roots. So if there is no growing roots there, some of this population will be affected of the mycorrhiza. So that's something which you need to make sure that everything is fresh and everything is alive. Mm-hmm. Usually I never trust the potting mix to be the sole source of mycorrhiza of our studies. We mm-hmm. usually applied fresh inoculum every cycle, and when you start it at early stages, the amount that is being needed for application is small. So it's really it's really doesn't worth taking the chances. You know what I mean?
Yeah, it's absolutely cost effective to apply it early in the plant's life because you don't need very much, and the biomass is going to grow along with the root biomass.
Exactly.
So that makes a ton of sense. And one thing I also see with labels is people will buy potting soil that will list mycorrhiza on the label, but in reality, the amount that's in there is so minuscule and small that they probably won't get infection, or if they do, it will take way too long. But what I'm relating it to is for people who do, let's say, some of these facilities that are growing cannabis in a raised bed will inoculate their plants in that bed. The plant will flower out in the bed, and then they will pull the plant and leave root fragments in the soil and then plant another plant back into that bed, another cannabis plant within 24 to 36 hours. And I'm wondering if doing this 5 times a year over the course of multiple years, if at some point there would be enough mycorrhizal hyphae or propagules that you wouldn't necessarily see a big difference in benefit in regards to direct application every time.
If you have the right strains, as I mentioned before, and probably you reach a level that there is no point to add more mycorrhizae in, then you really can. I don't know how many cycles you need in order to reach there, but biologically it's possible.
Okay, yeah, and one other thing that I wanted to highlight is, along these lines, from what you're saying, it makes no sense to just mix the mycorrhizal spores homogenously through our soils. It seems like that would be not very economical relative to applying it directly to the roots.
Yeah, that's what we tried. We call it we try to create a situation when we dust. You even can raise your let's say, if you missed it, then you are not applying it to the rooting plague in the beginning. Once you have the first, when you transfer it to the next stage, then you can dust off the whole how we call it chunk of root plague with mycorrhiza, and then make sure that every segment which is merging out of that plague to the soil somehow will get infection, and that's you can do it this way too. So you don't really need much for that. It's like if you dusting donuts with sugar. You don't need much of it. You just need to, and the rest or the excess amount you can kind of shake it. And it's really a minimum amount only on the surface, and you can make sure that you'll have again in the right concentration and really meet the terms of good colonization.
Now, one of the things that I want to talk to you about real quick before we get off the phone call was you helped create in a lab this particular super strain of mycorrhizal fungus that is used in the product Dynomyco, correct? Can you talk a little bit about that?
I can talk. I can talk about it in general terms, of course.
I know some of this stuff is proprietary, and I just mean in terms of you worked on this particular product and you saw some of the results around it. I was hoping maybe you could cover that, not necessarily the methodology, which you may not be able to share.
What would be the exact question? Yes, I know many of these. I know those strains, the superstars. There is more than one, but even the best one, of course, I'm familiar with that. And I think that that product came through a very intensive development, which is technically but also biological development, which brought it to the level that you can have millions and millions of spores, which are free of soil, of course, and you can really make sure that you have just mycorrhiza, and you can set it up in a way that you have in small volume a very high concentration of spores in a very not just efficient but a very effective way for infection. And that's something which is very important. They are actually giving you something which is maybe I should use which is being primed ahead of time and ready to go. And this is the advantage of that product. And and the results are nothing better than see the effect of the product on the plant is very convincing. And I think that the uniqueness of this product is many growers probably can tell you better. Remember that I never did it myself. Everything was done by application to cannabis was not done in our lab, but I'm sure that the company they check it and they try it, and the proof is in the pudding. I mean, that's working.
Great. Yeah, so this particular product was originally researched and developed out of the Volcani Center, correct? And then
Yes.
and then moved to a sort of a private enterprise. But
Right.
yeah, that was sort of what I was getting at. So thank you. One last question I had for you that was sort of unrelated, but one that I hear debated by growers that I think you could answer fairly readily is this idea of Rhizosphere pH and the importance of pH. Some organic growers don't really don't use pH as a tool because they say that the soil's ability and the microbes' ability to buffer is so great that it's less important. However, some of us, like myself, feel that while there is an aspect to that, by optimizing pH in rhizosphere and in the soil, we're allowing for the plant to have less stress and to make these make it easier for the plant to uptake nutrients.
Yeah.
What are your thoughts on this, and how do microbes help in sort of that pH and rhizosphere relationship?
First of all, microbes do have some effect on the rhizosphere effect, on the rhizosphere pH. But the most important mechanism that alter the pH of the rhizosphere is the plant itself. Mm-hmm. And in order to absorb the anion and cation, and there's exchange of anions and cations, the nutrients, the charge of the molecules is very important. And it's very important if you feed your plant with ammonium or you feed yourself with a nitrate. That makes a significant effect on the pH of the rhizosphere. So that's something which should be kept in mind. If you really want to alter the pH of your rhizosphere, work out the equations or the compounds, the chemical structure of the nutrients which you are using. If you'll have more nitrate, then your pH will level up. If you have more ammonia, your pH will level down. And that's more effect. This is more significant than the microbes themselves. And of course, the organic compounds which are being exuded from the roots are actually also a second layer of effectors on the pH itself. And that's something which we can barely control. So the question is, what's really happening in the if you have a lot of sugar, let's say, and you have a surface strain. I don't know if it's all the rhizosphere is covered with microorganisms, exactly like in our gut, mm-hmm. And you know that some of those microorganisms are responding to the given amount of the type of food that we are eating. In our gut. The same happening at the rhizosphere level. And if in our rhizosphere is going to exude some molecules, the bacteria there will transfer the molecules and can reach a situation that can influence the pH. And last and not least is the aeration in the rhizosphere. Do you have enough air, or you have anaerobic conditions? that will influence also the type of bacteria that can grow there, and will affect the pH. You have phosphorus. So on your soil, let's talk about sulfate. If you have a free sulfate somehow, you get a lower pH in the rhizosphere. So the chemistry and the biology also all the time is working, and I'm sure that you'll get a good recommendation if you have a good nutrient solution that will have high buffer capacity. Then you can eliminate those type of changes in the rhizosphere. It's very important, trying to understand what group of bacteria you have there or other microorganisms, and trying to influence this microorganism is kind of indirectly, it will, in conventional agriculture is almost impossible. So the only way to go is to have a nutrient solution which has a high buffer capacity that can work out the difference that the rhizosphere, the bacteria, and even some of the effect of the plant, which I described it, can buffer it up, and that's something which will be appreciated. This is a practical recommendation. Nowadays the microbiome is a new field that you can dive into this field, but it's very, very hard to tip it. Of course, if you'll put some compost in the roots, then you are going to change the flora, but also the conditions in the soil. Yes, pH is very important. Under high pH, if you really want to absorb, if you really want your plant to absorb enough iron for the chlorophyll to have a good color and photosynthesis in the leaves, you please make sure that the pH will not be above 7.5, even lower than that. 6.8 or something like this. If you have a low pH, then your plant will not be able to absorb some of the goodies that is being needed for a good crop. So this is something which should be kept in mind. My recommendation is take a nutrient solution with a high buffer capacity, and that will help you to overcome those situations in the rhizosphere.
So is it safe to say that the rhizosphere, plant rhizosphere interactions and the rhizosphere itself is highly complicated? We are still learning a lot about the soil microbiome in relationship to the rhizosphere. And by having the pH within a range that is more optimal for the plant, all we're doing is making it easier for the plant to access nutrients, which will potentially reduce plant stress and allow it to put more energy into yield and growth and protection.
Yeah,
those sorts of things.
Yeah.
Okay.
If you can measure it, if you can control it, under hydroponic conditions, you have more tools to deal with the pH. But if you have a potting mix, it's harder to start tipping the pH in the middle of the season.
Yeah, so the way that we grow is primarily in potting soils, organic potting soils, where we're using soil testing in between cycles to sort of, in some ways mimic, outdoor agronomic practices by managing that soil multiple cycles, but in an indoor environment. So we're constantly adjusting our nutrients that we're putting into the soil to maintain as optimal of a pH and mineral balance as we can, while still taking into account the biological aspects of the soil itself too. So.
Yeah, and of course you have to remember that you have to change your schedule, the nutrition schedule between vegetative growth and reproductive growth. You need different schedule there too. I mean, you cannot use one equation for the entire growth cycle for cannabis growth, right?
Sure. So the nutrients that the plant is going to want when it's in a vegetative state are quite different than what it's going to want when it's flowering.
Right.
That's what you're saying. Yes, yeah. That makes sense. Well, Dr. Kapulnik, I know I could I could ask you a million more questions, but I know it's getting late in the evening where you are in Israel, and I really appreciate you taking the time to share this information with us. Maybe we can chat again in the future sometime. I would love that.
Thank you very much. I enjoy it. It's a pleasure to talk about the rhizosphere. Not many people understand sometimes what we are talking about. And I will be very happy to contribute from our years of experience here to a new crop or a new industry that you guys are right now doing very smartly. And I'll be very happy to help you.
Well, thank you so much. You have a wonderful rest of your evening, and thanks for coming on the show today.
Thank you very much, sir. Bye-bye.
That was Professor Yoram Kapulnik, and you are listening to the Cannabis Cultivation and Science Podcast. Don't forget to check out the podcast page at www.kisorganics.com. Just click on the Learn tab, then Podcast. We have also added the Dino-Myco product that Professor Kapulnik worked on to our product selection, along with a bunch of other exciting new products. Thanks for your support and happy growing.